Hello. Hopefully we are live. Can everyone hear me okay? Give me a quick thumbs up or a hello if you can hear and see me. And also, where's everybody joining from today? Presuming this is working. And have we got any first timers, or have you guys been to these sessions before? Hello, Khadija, were you just in the GCSE class as well? Or is this a different Khadija? Aberystwyth, I can never pronounce that one. Aberystwyth, nice. That's North Wales, right? Hello, hello. From Birmingham, nice. Have we got any international students in today, or everybody UK based? I feel like Middlesbrough, nice. Northeast representing. It looks like the quality of the video is not amazing, but hopefully it's working all right. So we are going to go through respiration and glycolysis today. So this is a very important topic for biology. And actually, if you're in year 2, this is even more important than normal because this features, I think it's in all of the exam boards advanced information. So it's definitely in AQA and OCR, and I think it's also in EdXL. So yeah, it's a super important topic, and it's going to make up a reasonable percentage of your exams. If you're not year 13, to be fair, you probably wouldn't have actually really done this topic yet, so it might be a bit tricky. But by all means, feel free to try and follow along. But yeah, it is an A2 topic, what we're looking at today. It looks normal. Okay, that's good. Glad to hear. Thanks for clarifying. So yeah, before we start, a little bit about me. My name is Alex. I'm the head of biology here at SnapRevise. I've been teaching and tutoring biology for the last 6 years now. And yeah, I did biology at university and I was working in schools for a little while as well, but now just doing tutoring. So yeah, a little bit about what like some courses we have. So I don't know if you guys are aware, we ran these seminar courses, the sort of crash course for biology, chemistry, physics, and maths. We did these over the Easter holiday. So that they've already happened for this year. So unfortunately, you can't go to them live. We're not doing them again, but you can access the recordings. So we've recorded everything. We've got all of the booklets as well. And you can actually, although that was in the past, you can still message the tutors to ask questions. So that functionality is still going. So if you missed it, no worries. You can basically get nearly all the benefits by downloading these packs and accessing the content from the recordings. So yeah, check them out. The link is in the chat now. So yeah, on with our session today. So we're going to recap a little bit. This is kind of a learning pyramid. We use these at the start of the lessons when we do our web classes normally, just to refresh some of the ideas and to look at some of the base knowledge you need to access the session properly. So let's go through. First question, what is respiration? How would you define that? Mm-hmm. Yeah. So it doesn't just have to be glucose, but that's usually what people tend to say. But probably better to say use biomolecules, because you can also respire proteins and lipids. So use biomolecules to synthesize ATP. Something like that. Yeah, so you're kind of right. Like glucose is the most common respiratory substrate, but it's not the only one. So you can talk about other stuff as well. Okay, what is the symbol equation for respiration? Oh, Zara, you've got it kind of on there already. Yeah, I guess it didn't actually specify here. This would be like aerobic respiration. So you have glucose plus water, sorry, plus oxygen, goes to CO2 plus H2O. That's not, oh, sorry, we're doing the symbol, aren't we? Yeah, okay. So the chemical formula for glucose, C6H12O6, and you have 6 of all the other molecules. Yeah, nice, exactly. I think everyone's got that. Everyone's got that correct. Cool. Perfect. So where does respiration occur? We can split this into aerobic and anaerobic. Yeah, it occurs in all cells. Nice. And then specifically in those cells, aerobic is in the mitochondria. Yeah. And anaerobic, cytoplasm. Yeah, nice. Cool. Then next question. So what is the difference between NADP and NADPH? This is actually photosynthesis rather than respiration, and it's because we normally do photosynthesis before this lesson. So it's following on from a previous lesson, but obviously we haven't done photosynthesis, but hopefully you guys will know this topic already. One is reduced. Which one? Well, actually, I've kind of drawn a line to it now. Yeah, so this is reduced. And that means it has, yeah, gained hydrogen and gained electrons. Gained H plus and gained E minus. Nice. And so this is a coenzyme and it transports these molecules from place to place in the cell. So yeah, very useful. There's also in respiration, we're not going to look at them today particularly. Well, actually, we'll look at NAD a bit. And there's also FAD. These are the respiration ones. And they can be reduced like that. And the reduced one, you just write an H to show that it's reduced. So how does RUBP get turned into GP? Again, this is photosynthesis content. Catalyzed by Rubisco. Nice. Yeah, the step is called carbon fixation. And basically 5 carbon RUBP plus 1 carbon CO2 get combined to make a 6 carbon molecule. Some specs, you need to know the name. Actually, no, for this one, actually, no, sorry, no one needs to know the name for this one, just a 6 carbon intermediate. And this then splits into 2 3 carbons. And these are GP, basically. So 2 times GP. That happens in the stroma of the chloroplast. So that's like the equivalent of the cytoplasm inside the chloroplast. Nice. And then final question. What's the structure and function of ATP? We're not looking at the link reaction today, actually. We're just doing anaerobic and glycolysis. Yeah, it's often called the universal energy currency. Nice. Yeah, exactly. So an adenine base, which is a purine base, so it's a double ring structure. And then it's a pentose sugar, which means a 5 carbon sugar. Specifically, it's ribose. And then there's 3 phosphate groups. So it looks a bit like that. So it's actually based on an RNA nucleotide, hence why the sugar is ribose. Because if you got rid of these, it's basically an adenine RNA nucleotide. It's a small difference, but that's the idea. Nice. So let's go through. So that's the main objectives today. I think we'll have time to cover all of it. We'll see. It usually takes a little bit longer on these YouTube ones. So these are your spec points. You should all know your spec inside and out, particularly if you're year 13. You should have spent quite a while looking at them already. So yeah, this is a copy in case you haven't seen it, or just to show you, to remind you of this particular part of the spec. But yeah, you guys should all have this already. If not, you can download them really easily. Yeah, sometimes they get fussy about that to like make energy, because it's less of an issue for biology. I think this is where physics, they really are like quite hot on this. But yeah, technically you don't make energy, you change energy from one form into another. So yeah, better to say you synthesize or produce ATP rather than, yeah, you can't create energy from scratch. You only ever change the form of energy. But yeah, the physics are more fussy on it, but that's still good to get in the sort of the habit of using that terminology. So we said ATP acts as a unit of energy. And yeah, basically it can be hydrolyzed to ADP plus PI. This stands for inorganic phosphate. Hydrolyzed to this, and this releases energy. And this can be used for lots of different processes in the cell. So what are some of the things we might use ATP for in the cell? what processes can you think of? There are quite a few that you will have covered during the syllabus. I suppose we use them in glycolysis at the start, but I probably wouldn't go for that. Protein synthesis, nice, yeah. Muscle contraction, that's probably one of the biggest ones, yeah. Sodium, potassium, presuming that's meant to be pump, yeah. That's a good one. Nice, exocytosis, good. Calvin cycle, yeah. So in photosynthesis. Yeah. Oh yeah, not the Krebs cycle. Krebs cycle actually produces it. Mitosis, yeah, for sure. You need quite a lot of mitosis. Nice, and obviously also meiosis and also endocytosis as well. Yeah, perfect. Active transport, yeah. So this pump is one example of active transport, but obviously there are other examples that you can have as well. Cool. So let's look at this overview then. So this is showing sort of the whole of respiration in a very simplified form. So you'll notice there's two branches. You've got anaerobic and aerobic. Aerobic, kind of as the name sounds, requires oxygen. So requires O2. Anaerobic does not. Let's put no O2. So there's two different pathways, but they actually both start with the same thing. They both start with glycolysis, which is where you turn glucose into pyruvate. And we'll look at how that works. There's a few steps in it. So glycolysis is always first, and then you can choose whether to go down the anaerobic pathway or you can go down the aerobic pathway. If you have oxygen, the aerobic pathway will always be used because firstly, it makes a lot more ATP. So for this one, you only get net 2 ATP per glucose. Whereas this one, you get, the numbers vary a little bit because there's some inefficiencies and some wastage along the way, but a theoretical maximum of 38. But sometimes when you factor in the waste, sometimes you see this number down as low as 28. But let's put 28 to 38 ATP per glucose. So this is a huge amount more. It's not even really comparable, like over 30 versus 2. So you get a lot more ATP. So this is definitely preferable in terms of the quantity of ATP produced, but it's also preferable in terms of the waste product that you produce, because the two waste products, one is water, which is totally fine. We have loads of water in our cells anyway, so that's not even really treated as waste. It will just be incorporated into the cell. And the other waste product is CO2. And this one can cause problems. If the CO2 builds up too much, then it will lower the pH, and that can actually eventually denature proteins and enzymes. However, the body's got a very good mechanism for removing CO2. We just put it back into the bloodstream and we exhale it, so we get rid of it. So the waste products aren't that bad from aerobic. The CO2 is easy to get rid of. So this is always preferable. However, if you don't have enough oxygen, that's when you'll do anaerobic. And that's what we're going to focus on today. So the anaerobic pathway. So starting with glycolysis, and then assuming there's not enough oxygen for aerobic, we go down this pathway. Then there's two options. Humans or mammals, we produce lactate or lactic acid, whereas yeast and plants, they produce ethanol and CO2. So you can, we'll look through those two different pathways in a sec. So hopefully that's making sense. we've already done this before, the similar equation, so let's write it on quickly. C6H12O6, but this should be like second nature for you guys by now. Oops, I struggle to talk and write at the same time. CO2, H2O. So again, 6, 6, and 6, and 1 glucose. So yeah, this is a mitochondria. There's a few features. It's got a double membrane, outer and inner. This is actually very important because you build up a gradient of hydrogen ions in between the two membranes. Then, yeah, other features, the matrix. This is like the cytoplasm. Cytoplasm of the mitochondria. And the different stages take place in the different areas. So in the cytoplasm, or i.e. the matrix, this is the link reaction and the Krebs cycle. And the inner membrane, this is oxidative phosphorylation. And glycolysis actually takes place outside of the mitochondrion, so that it doesn't go on in there. Yeah, 666. Is it like the number of the devil, right? I don't know where that comes from though. But yeah, use it to help you remember it though. So how are we feeling with that so far? Nice. Perfect. Yeah, let's go through some questions then. So these ones are not too tricky. So where does glycolysis occur? And where does the electron transfer chain take place? If you didn't remember, this is part of oxidative phosphorylation. OXPHOS for short. Cytoplasm. Nice. Inner mitochondrial membrane. Yeah, perfect. Then have a look at this question. So 2 marker, why do mitochondria in muscle cells have more cristae than mitochondria in skin cells? ETC is on the inner membrane. That's kind of like embedded into the actual wall of it. Yeah, you get a greater proton gradient. So that's because of the increased area for electron transport chain. So cristae are folds in the inner mitochondrial membrane. So same thing as this. So increased surface area for inner mitochondrial membrane. Then yeah, something like more enzymes for ETC. See? And you could therefore say like more ATP generated. Nice. So let's actually look at glycolysis in a bit more detail now. So we said this is the first stage of aerobic and anaerobic. So essentially what you're doing in glycolysis is splitting a hexose sugar into two smaller molecules called pyruvate, which are both three carbon molecules. So a hexose sugar, hex, tells us that it's 6 carbons. So the hex part refers to the number of carbons, just like a pentose sugar or a triose sugar is 5 and 3 carbons. And then the O's part, this tells us it's a carbohydrate. So this is kind of useful, just like how if something ends in ASE, you know it's an enzyme. If something ends in OSE, you know it's a carbohydrate. So it's just a useful thing to know. Yeah, good question. So cristae isn't actually the name of the inner membrane. Cristae is the name of the folds in the inner membrane. So if you looked at a mitochondrion, the inner membrane is folded like that to increase the surface area. So cristae is literally the folds in the inner membrane. Whereas it could be like unfolded, and this is going to have a smaller surface area because all the enzymes are embedded in this. If it's not folded, you're going to have way fewer enzymes than if you have folded, and there's like enzymes the whole way along. To be honest, like for mark schemes, you could almost use it interchangeably because they usually accept inner membrane or cristae, but technically the cristae is just the folds. But yeah, the way the mark schemes work, it almost looks like a synonym. Yeah. The matrix is the rest of the fluid. Yeah. So the matrix is in here. And the gap between the two membranes, this is your intermembrane space. This is where you get the proton gradient, which generates the ATP in aerobic. But we're not doing aerobic today. So this is a 3 carbon molecule. And can enter either aerobic or anaerobic pathway. But depending on O2 availability. Cool. So let's go through the steps in glycolysis. So there's 4 main steps you can break this down into, and we'll go through each one. There actually are enzymes that catalyze every stage of this reaction, but we don't need to know the names of any of these, which is nice. And actually, to be fair, the A level simplifies it down to 4 steps. It's more like a 12 to 15 step pathway, but a lot of the reactions, not that much happens. So we've ignored those for A level and we just focus on the most important ones. Yeah, luckily they decided that just learning loads of different random names of enzymes wasn't the best test of your biological knowledge, which is a good thing because it doesn't really, I mean, you could learn them, but it doesn't really help you understand it any better. So yeah, they've got rid of that. Oh, I say got rid of it. I don't think it was ever in there, but there is basically enzymes that are involved. So the first step 1, phosphorylation of glucose. So this means adding phosphates to glucose. Yeah, at, at uni, you do have, well, at least the course that I did, you had to learn all the steps and the enzymes, which even at uni, it's kind of pointless. It doesn't really, again, it doesn't really test your knowledge, it just tests your ability to learn a pathway, which if you ever needed to look it up, you could look it up in 2 seconds. So yeah, I don't know. Anyway. Yeah, step one is adding phosphates to glucose. So this is actually a bit weird because the whole point of respiration is we're trying to make ATP. And in this first step, we're actually using up ATP. So we're using 2 ATPs and we're taking the phosphates and we're sticking them onto glucose. That's the blue dots that appear. So actually we don't get any inorganic phosphate because they're stuck onto the glucose. That creates hexose bisphosphate. There's an S missing from here. Hexose bisphosphate is unstable. That's why we put the glucose on, to make it unstable. Because it's unstable, it splits into two times 3 carbon molecules. Yeah. Depends on your exam board. What's that for? Oh, whether you have to know about that. Yeah. I think even for AQA might talk about hexose bisphosphate. But yeah, it's not the most important step. Like phosphorylation to TP. Yeah, there's not much to say about hexose bisphosphate other than it's unstable. So if you've learned it with splitting that one out. Yes, there's quite a few other names for it actually, annoyingly. Some specs call it phosphorylated glucose. Some call it phosphorylated fructose. Some call it, I think that's the main ones. Put it here. Phosphorylated glucose or hexose or fructose. I've seen all of those words instead. Oh, there's another word, hexose phosphate. Yeah, hexose phosphate would imply that there's only one. And actually, when we do this question later, so hexose phosphate is actually one of the extra steps. So between glucose and hexose bisphosphate, you add 1 ATP and you get that. You add another one and you get bisphosphate. So that is actually another step. Sometimes they put it in, sometimes they don't. So we've split our hexose bisphosphate. Now we've got our 2 3-carbon molecules called triose phosphate. This is also known as TP, often, just for short. And it's the same thing that you'll come across in photosynthesis. So maybe you recognize it from that as well. So our TPs, we're going to oxidize them. Oxidation and reduction always happen in tandem. So when one thing is oxidized, the other is reduced. Is this frozen? There seems to be like a huge lag on this. But hopefully it's working. Okay. Yeah, I think it's like almost 20 seconds behind when I'm saying something and when it like appears on YouTube. So hopefully it's still you can follow along okay. But I guess I'll get your answers quite a long time after you type them. or quite a long time after I ask a question. Hopefully it catches up a little bit. So oxidation of triose phosphate. This is important because we have these things called redox reactions. This is where one thing is oxidized. So the ox part is oxidized and another thing is reduced. So if you have an oxidation, something else needs to get reduced. So we oxidize triose phosphate. Oxidation, you guys have probably come across oil rig before. I mean, almost definitely. Oxidation is loss. So lose hydrogen plus electrons.
Sorry, I'm having trouble understanding right now. Please try a little later.
So lose hydrogens and electrons. This at the same time, NAD is reduced to NADH. Then we produce, I should say, pyruvate. So at the same time, the TP is being oxidized to pyruvate. We actually make some ATP now. So ADP is phosphorylated. To ATP. So this actually happens, there's two of these per glucose. So 2 times this, 2 per TP. So overall, we make 4 per molecule of glucose. So 4 ATPs are produced. However, we used up 2 in the beginning, this first step. So total 4, but net 2, we often say, because 2 were used up. So be careful with your questions. Sometimes they ask how much ATP is made during glycolysis in total. In that case, you say 4. And sometimes they say how much is made kind of overall or net. And in that case, you would say 2, because you factor in the 2 that have been used up. So just pay attention to that, because they try and catch you out with the questions. Okay, so yeah, let's keep track of what we've made. So this is what we've made in glycolysis per molecule of glucose. The grade boundaries, yeah, I don't know. Actually, I think they'll be higher, to be honest, because of the advanced information. There's less topics to revise. So I think grade boundaries are always relative. They can't give too many people A stars, and they can't give too many people A's. So they've kind of got rough criteria. Like maybe it's like 10% of people get A stars and 25% get A or A star. So like the first top 10% get A star, the top next 15% get an A, and so on. So if people do better, the grade boundaries go up. And with the advanced information, in theory, people probably will be doing better because there's less to revise. Yeah, they were talking about either lowering the grade boundaries or this like advanced information. And yeah, it's all a bit of a mess, to be honest. Like, we don't know until the actual exam comes up how much you can trust the advanced information. Maybe there won't be any stuff that's not on there, but some specs have been quite vague about it. So yeah, it's a little hard. It's all a bit of a mess. Feel bad for you guys if you're sitting your exam this year. It's been, if anything, more stressful with the advanced information, I would say. Yeah, I guess we'll see what happens. So yeah, so what have we made so far? Net 2 ATP. Nice. Yeah. Yeah, they said AQA particularly said here are some topics, but yeah, obviously anything can feature in the essay. Although to be fair, you can kind of pick and choose what you talk about in the essay. So if you haven't revised a topic, it's unlikely you would actually need it specifically. You can talk about something else in the essay. Yeah, 2 of these and 2 of these. So it's actually nice and easy. Just 2, 2, and 2. If you're going for net, so nice and easy to remember. So let's try a question again. So what's the name of this stage? Mm-hmm. Nice. Yeah, I think they did that to keep their options open, just saying that they could come up in shorter answer questions in case they put some stuff in. But you don't know how, yeah, it's tricky to interpret. So it's probably worth reading over the stuff a little bit, that even that's not mentioned, so you can approach the shorter answer questions. Yeah, cytoplasm. And then what we're looking at. NAD, nice. It's actually not reduced NAD, just NAD, yeah. Because it says reduced here. F is pyruvate, nice. And yeah, ATP, nice. Cool. So let's go through anaerobic respiration. So glycolysis, that happens regardless of aerobic or anaerobic. And then we do the anaerobic pathway if we don't have oxygen. Did these papers get leaked? I didn't hear about that. Wow, yeah, that's a bit of a problem. I was going to say, because they normally have a reserve paper, but they didn't have a reserve this time. I don't know what to even do then, because you can't just do the exam. Really? That's interesting. Yeah. I literally don't know what you'd have to do, because even if you had a backup paper, it's quite hard to get the paper out. So somebody's going to get fired, probably. I don't know who leaked it. It's amazing it doesn't happen more often. There's so many potential points that papers could get leaked, because loads of people handle them. Like, firstly, everyone who made the papers, they know about them months in advance. And the people who work in the printing shops that print them out, they know, they've obviously seen the papers, and then they get sent to all the schools at least like a week before the exam. So they then sit in a school office where there's quite a few people who have access to them. And there's loads of schools in the country. All it takes is one person to like look at them and leak them, and that will spread, because, well, on the internet it'll spread so quickly. So yeah, I'm amazed it doesn't happen more often, but it seems to. Yeah, you have to have a safe. Yeah, every school has to buy a safe to put the exams in, but who knows how many people have access to the to the safe. Yeah, I'm always surprised it doesn't happen more. Maybe it does. Maybe it's just not shared. Anyway, hopefully it won't happen with the A-level paper, because I feel if anything, it'd be more faff. They'll have to just do another way of examining. So this is kind of a misconception that a lot of people have. Like, what is anaerobic respiration? It doesn't actually make any ATP anaerobic respiration. What makes ATP is glycolysis. And then anaerobic respiration, this only allows the replenishment of NAD, i.e. turning NADH back into NAD. And the reason we do that is so we can do more glycolysis. So this is what makes ATP. It's net 2. So we want to do more glycolysis. However, glycolysis needs NAD, and there's not that much NAD in the cell. So if you want to keep glycolysis going, this is glycolysis, where we go glucose to pyruvate. If you want to keep glycolysis going, you need more NAD. So anaerobic respiration just regenerates NAD from reduced NAD. And then this allows, so we need NAD. This allows this last step, TP to pyruvate, to carry on. So that's where we make our ATP. So yeah, NAD is super important. So we regenerate it by turning pyruvate into lactate in mammals. So essentially, lactate is just reduced pyruvate, because we said these redox reactions, if one thing is reduced, something else is oxidized. So if pyruvate is getting, if NADH is getting oxidized, pyruvate is getting reduced. So lactate is just reduced pyruvate. Yeah, exactly. So reduction is just the gain of hydrogens and electrons. So at the moment, the NAD is carrying hydrogens and electrons, and it's just going to give those hydrogens electrons to pyruvate, which then turns into lactate. So pyruvate is just reduced lactate. Sorry, other way around. Lactate is just reduced pyruvate. No, it doesn't get decarboxylated. That's what happens in yeast and plants. We'll look at that one next. In this case, lactate is still a 3-carbon molecule, and this is a 3-carbon molecule. So we haven't lost any carbon, so it's not been decarboxylated. So we're looking at some more notes. In the absence of oxygen, no Krebs or link reaction or oxphos. So only glycolysis makes ATP. So we need glycolysis to keep going. NADH is oxidized back into NAD to allow more glycolysis to occur. And yeah, generate more ATP essentially. Yeah, exactly. And this product is actually toxic. So lactate, i.e. lactic acid, this builds up, and this is actually what causes muscle cramps and that feeling of like the burning sensation when you're exercising. If you're exercising intensely, like a sprint or anything that you're exceeding your aerobic threshold, i.e. you're starting to do anaerobic respiration. Anytime you exceed that, you start to do anaerobic and you'll feel the lactic acid accumulating. So it's uncomfortable. It's like burning your muscles. Yeah, exactly. So this is why you can't do anaerobic forever, because you're going to eventually have too much lactic acid, and yeah, your muscles just cramp up and they stop working. So first it's just uncomfortable, but eventually, even if you're like super disciplined and you can push through the pain, eventually the muscles just stop working. They get weaker and weaker. So you can't respire anaerobically for forever. So we can write this as an equation, maybe a little bit differently. Oh wait, that's the ethanol one. So just pyruvate plus NADH goes to lactate plus NAD. We don't make any ATP. We've just regenerated NAD. That's the whole point of this. So yeast and plants, they do it a little bit differently. They turn pyruvate. Well, what do they turn it into? Yeah, some specs you do it into two stages with ethanol first. I'm just going to do it in the simpler form. Ethanol plus CO2. Yeah, and we could either draw in the NAD and NADH, or we could add it like this. So in this case, 3 carbons, 2 carbons, 1 carbon. So in this case, pyruvate is decarboxylated. Because decarboxylation has occurred, this reaction actually isn't reversible. So you can't turn ethanol and CO2 back into pyruvate. In the mammal one, you can actually turn lactate back into pyruvate. And that's what you do. After you finish respiring anaerobically, you've got all this lactic acid and you have to convert it back into pyruvate. This actually requires a lot of oxygen. And this is why you keep breathing more intensely for quite a while after you exercise anaerobically. It's called the oxygen debt, and you're repaying the oxygen debt. So essentially, you're just turning all this lactate back into pyruvate. Plants and yeast, they cannot do that because it's lost carbon. And it's actually very hard to turn inorganic carbon back into organic carbon. So the only step that does that is the one we looked at on the very first page, where carbon fixation in the photosynthesis cycle. Yeah. So if you were growing some yeast, so we do this, we use this fermentation effect to make alcohol. To make this ethanol is the same alcohol that gets humans drunk. Actually, not just humans, it's any mammal. So you might have seen videos of sometimes elephants, they eat fruit that's been fermenting on the ground and they can get kind of tipsy. Same with like wild boar, they sometimes eat fermented apples, kind of also almost like cider, and they can get pretty boozed up as well. So it works on other mammals. But yeah, the reason they're doing that, the yeast are doing it, is purely to make the ATP from glycolysis. And they then regenerate their NAD by turning the pyruvate into ethanol and CO2. So these are waste products for yeast. They don't need ethanol for anything. CO2 also a waste product. And in fact, the ethanol is toxic for the yeast. So they can tolerate a certain amount, but they can't have too much. And that's why when you're fermenting something to make an alcoholic drink, you can, so something like beer or cider or wine, the maximum strength you can get to is around 14 or 15%, because after that, the yeast die. So anything that's just purely fermented, i.e. beer, wine, cider, mead, although no one really drinks mead anymore. These are all limited to how much ethanol the yeast can tolerate. So it's maximum around 14.5%, 15%. If you want to make stronger stuff like spirits, you need to distill the alcohol, and that's where you just concentrate it. But the yeast, you can't like just make vodka or tequila directly with the yeast. You need to distill it. What was the question? Glucose phosphate. I think someone's answered it exactly. You need to add another phosphate to make it bisphosphate. Then what other question? How the NAD is going to NADH going to NAD. So this is being oxidized, and the pyruvate is being reduced. There's basically hydrogens on the pyruvate. Sorry, hydrogens are on the NAD, hence the H here. And this is being given to the pyruvate, and that's what reduces it. Yeah. So we've got a couple more minutes. Let's just do a few more questions, and then we are basically finished. So that's actually worked out quite nicely with timing. So let's try these questions, and then we will end there. On the slide it says pyruvate is oxidized. Yeah, that's a mistake then. Yeah, you're right. Reduced. Well spotted. I've actually never noticed that before, and I've used this slide a lot of times. So, nice. So first one, cytoplasm. Yeah, true. Then, what's the difference between oxidation and reduction? Yeah, so always refer to oil rig for that. Oops, what am I doing? Rig. So oxidation, reduction is loss and gain of chemistry. More talk about electrons. To be honest, for photosynthesis and respiration, I would think of it more as from hydrogen, but it's both. And then, yeah, so some good answers coming up for this one. So it regenerates a NAD. Which can then be used to accept either hydrogen or E- in glycolysis. Or you could say TP to pyruvate, because that's the stage that it's needed for. And this allows the body to, or allows the cell to continue to do glycolysis, which allows it to make ATP. So yeah, that's the main idea there. Then yeah, that's it. So hopefully now guys now know the basic sort of overview of aerobic. glycolysis and what we actually do in anaerobic respiration. So I'll stick around for a few minutes. If you've got any questions, I can answer those questions. Don't forget, we've got the crash course recordings, so check them out. I think we'll have the link again in the chat. If not, it's right at the beginning of the session. So check those out and see if it's, yeah, they're super helpful. So I would say definitely worth getting those. Yeah, so well, just a little bit on what we actually went through on those. So it's tailored specifically for the advanced information. So yeah, it's exam board specific. We go through some of the questions. We go down, like break down how you actually approach longer answer questions. We look at command words, that sort of thing. Then we look at some of the more challenging topic areas. So actually we do respiration in those crash course sessions. And yeah, you get this very handy booklet. I've got a copy of one of them here actually. So this is all the slides. So you get a copy of that. And yeah, you can still message the tutors to ask questions. So another useful little feature. That's the address, but yeah, it's also in the chat as well. Cool. So what questions have we got? What's the next session? Not sure what topic we're going to do. It's going to be next month at some point. But yeah, I think we haven't decided a topic yet. Or check the schedule on YouTube because maybe it has already been selected, but not sure. Pyruvate moves to the mitochondrial matrix by active transport. So that's actually one of the times that we lose ATP. So yeah, it's an active process. It actually uses up ATP. Explain the Krebs cycle. Unfortunately, I haven't got time to do that now. That would be like a whole 10-minute sort of explanation. But we've got some other sessions. I think if you go back to the archives of these live streams, I'm pretty sure we've done one on the aerobic pathway at some point before. So you can check that out. If you're a molecule in glycolysis, which one would you be and why? I love the abstractness of the question. I don't really have a good answer for it though. Yeah, no, I guess glucose tastes nice. It's like sweet, so I'd go for that. Glucose is a good one. I actually don't know whether you can buy pyruvate directly and like eat that, but I imagine it would also maybe it'd be all right. Recombinant DNA. Yeah, I can put in a request if it's not been decided. Yeah, I think that's a topic that lots of people struggle with, so that would be a good one to do. Sweet. In that case, yeah, thanks for joining. Thanks for yeah, giving up your Sunday evening to learn. It's a good sign that you're dedicated to your studies. And yeah, hopefully see you guys in the next one. Or if you enjoyed the session today, we do these classes 5 times a week on our website. So that's a subscription-based service. So check out snaprevise.co.uk and have a look because yeah, we do a lot more of these. We only do one a month on YouTube. We do like 25 a month on our website. Glucose is unstable. No, phosphorylated glucose is unstable. So yeah, I guess pick that one if you're feeling a bit unstable, which I imagine coming up to exams, maybe everyone's feeling a little bit shaky. Cool. So yeah, see you guys in the next one. Bye. Bye-bye.